Oopsie daisy. I went ahead and flipped Mark's switch, so he's really grumpy now.
I am.
Parental guidance warning.
Now keep in mind that I've been doing
extra grumpy. Don't watch this if you're in a good mood.
Yeah, because if you saw the last video, I'm assuming these are in order, but you know,
we sort of
sometimes you try, sometimes you don't.
Sometimes I can't.
But you know, we were talking about pain and how it's sort of like it looked like people are on the right track on getting down to a surgical method for solving what's called intractable pain.
Intractable chronic
people have
pain.
chronic pain. They take morphine and they can't get rid of it, and they're in terrible pain.
And I can tell you, because I have visited this place and I have lived there for many, decade or so. It is living hell to have pain that you can't touch with anything. So,
but the thing that gets me riled up is looking at, wow, there was a series of papers. And this taps into a lot of grumpiness on both our parts. Number one.
This goes right to the wellspring of our rage.
Now, we had talked about the doctoral student who educated us on a few things. And
yeah, good stuff.
Because, quite frankly, we don't have time to read all
he's
the
a good
literature.
scholar, right? So that's the thing. And a lot of it, he was digging out papers that are 140, 150 years old. And Mark and I love that because some of it's very, very, it's just unvarnished, very revealing, very simple instruments.
Tells you what people were doing and gives you a lot of history.
And it gives you the perspective that not all these ideas are new.
Actually got comments from a supposed co-author one day recently who didn't like an intro that we had crafted that went back to 1860. What are you doing all this history for? I'm like,
right. He means comments on a scholarly paper submitted to a peer-reviewed journal.
Well, actually, this was just submitted to a supposed co-author.
Submitted to okay.
Yeah.
Right.
Anyway, wait a minute.
What are you doing all this? Yeah. What are you doing all this history for? Because the present is based upon a foundation of everything that happened in history, and in particular, science is history, right?
But so anyway, the thing that when you read the history, you can look at trends and you can see, wow, these guys are on the right track. Mm-hmm. Go back and watch the last video if you didn't watch it. But they're on the right track to saying what part of the brain could you ablate or modify to get rid of this intractable brain.
Where in the brain might there be, whether you want to go in there and cut it or you just want to study it some more to understand it.
And my mentor actually wanted to do some localized pharmacologic.
Another thing you can do are non-invasive ultrasound, right? And you can get, they do it once or twice and they do get some semi-persistent relief.
And TMS as well.
TMS. So you get, if you can stimulate by, some means that's acceptable to you as a person, that particular point on your somatosensory cortex, somewhere around what's called the central sulcus. Is that correct?
It's a central sulcus.
You can actually suppress that maladaptive pain.
Yeah, and there's several things that are going on. One part of that brain, it gets hyperactive, and it gets so hyperactive because it has to do with an acute to chronic transition. So acute pain is where, if I hit Bob in
which he does
slash him in the
time.
arm or hit him in the arm really
I'm
hard,
a victim.
he'd say, "Oh, that hurts." Well, if I kept hitting in the arm, or if it was a real injury and it lasted a long time,
then you go from
acute to chronic because you got signals going both directions.
So now your brain's kind of, I would say, actually, that's kind of a brain plasticity thing, right?
It is, and plasticity is very necessary.
I'm used to giving you a pain here instead of saying, okay, that's a signal I'm getting, and I'm going to stop interpreting it as pain. Your brain starts to say, well, any signal I get from there now, I'm associating with pain. Right?
Yeah, we call it maladaptive plasticity.
Maladaptive plasticity.
But we've talked about excitation and inhibition, and one of the big players in inhibition is called GABA, and GABA is what we call it's an inhibitory neurotransmitter, and it suppresses. So GABA plays a big role in suppression, but sometimes it becomes excitatory.
Right.
It flips in these maladaptive situations.
Biphasic response.
Yeah.
Right. So.
But anyway, that's the longer part of the story. But the big part of the trend that gets me riled up was looking at something where people were on the right track. And in 1969, a couple of supposedly prominent guys came out and said, "Bah humbug.
Right.
This doesn't work."
Well, this relates to Mark's recent and past experience with discoveries in areas of the brain.
And so in this same area in the brain, we did some studies, and I was mean, this was like my postdoc years, or just
that must have been
afterwards.
hundreds of years ago.
Yeah, like 50,
When
60
you were
years
young.
ago. I was young and not.
Back when you were young.
Yeah, I was very naive and not very grumpy. But
I used to
imagine,
think that science was pure. Somehow.
The thing about this. Okay, so those guys in 1969 shot everybody off into the wrong direction, and that sort of got, so everybody forgets the history and they forget their lessons and they move on. So all of a sudden they're doing incremental research in a different area of the brain, and they're all sticking electrodes in one place in the brain. It's actually called 3B1.
3B1.
Okay, so they're sticking electrodes. And there was one guy in particular, he spent 30 years sticking electrodes in that part of the brain, saying, well, that's where pain is.
How many data points did he get
Got 18.
30 years?
Got about 18
18 data points in
And
30 years.
so,
okay, that's like every other year. Oh, I got a little data blip.
Yeah.
I'm going to write that one down.
Yeah, I'll go
and then like 2 years later, oh, I got another one.
Well,
not really a juggernaut steamroller.
So,
of data
you
collection.
keep in mind, they're sticking electrodes in one specific location. We go in, open up everything, say, we're going to image this at really high resolution. I don't know. I'm not going to pretend I know where it is. And, I actually talked to people who said, well, you should just be focused on this area. I'm going, that doesn't make sense. We should look over here. So we look at the whole thing, and I say, hey, look what's lighting up.
Well, that's the thing. So he's looking at a larger area of the brain that includes, instead of 3A, which is somewhat adjacent, sort of. Close.
Yeah, it's next. It's right next to 3B.
Okay, so and then all of a sudden he sees all this activity.
Yeah, all this activity in response. This is activity that's related to pain. Is over
noxious
here.
Stimuli, I think.
Noxious stimuli. Yeah. And it was like, hey, this makes a big difference. So we say, publish that paper and say, wow, I guess people are going to get real excited about exploring those interactions and talking about.
So he's looking at the entire telephone exchange and saying, no, it's actually these telephones over here that are ringing when you call them, not the ones you've been studying for 30 years.
And so maybe if we studied that area, we'd have some way to think about diminishing pain or ablating it or something. And you look, certain stimuli actually reduced activity in 3A, which also perceptually reduced pain. So we had all this data go in, but guess what? The head of the study section.
Strong. So the study section, what is that? That is the group of senior scientists at the NIH who review impartially all the grant submissions. They decide which ones are good and which ones are not, and then they rank them.
Yeah, and the head of study section has a lot of power. He decides who gets to review what.
Who gets to review what.
He puts in his own opinions.
And then how the reviews, that's the whole tone of the reviews, and a big influence on the order of things. And when you're only funding maybe a third or a quarter or 10% of the grants that go in, boy, you lose a spot or two on the rank list, and that's instant death.
Yeah, but well, guess who?
It's called being blackballed.
Guess who the guy head of study section was one of these guys. He spent 30 years studying one place in the brain,
18 neurons,
and refused to say that this was
he kept studying the same thing, expecting to finally one day prove that he was right. You see this all the time in medical research, and he just wasn't. He would occasionally get spurious data, in my opinion.
Trashed our grants, trashed papers, personally trashed me and papers, and then a couple years later, he did his own study, found out, lo and behold, and he discovered that area became active with pain.
The area that Mark had discovered.
He did not cite our work.
Submitted.
And then he took off. Then he retired.
That he had confidential privilege to review Mark's work prior to funding or publication. He went ahead and just stole it and published it.
We published. He just didn't publish. He did not cite our publication. You're getting that confused with another guy who stole our stuff.
That's right. I've heard so many of these stories.
There was another guy that stole our stuff.
Grumpy mind, and just everything is just all soiled, right? But this was a case where they actually were able to publish, but they weren't able to get grant funding. And then one of the people who had a confidential access to his proposal went ahead and then more or less did the experiment.
Well, he did the experiment, but didn't cite our work.
Didn't cite your work. So he's trying really, really, really hard to hold people down. And this is really remarkably common.
Basically called these guys the pain mafia because they sort of had the NIH by a stranglehold. And if pain research did not go in a particular way, if your research didn't go in a particular manner, you weren't You were just not going to get funded.
That's right. And I mean, I could go on and tell grumpy stories, but if I do,
There's
Mark will ban this video.
There's probably a tissue engineering.
You know, the strangest one, there's this kind of mafia in all science. Did I tell you about my optical distrometer?
No.
An optical distrometer is a device that I invented with a colleague of mine named Neil Cole back in 1996 to measure atmospheric precipitants, like rain versus snow, and all those wonderful, colorful images you get now in radar, like weather radar, are because 25, 30 years ago, we were actually, well, not we, other people were actually measuring what was the actual precipitant coming down. Was it a raindrop or a sleet or hail or snowflake? And then correlating that with the remote sensing from satellites and stuff, or radar, and then they were able to model it well enough to be able to say, oh, this is snow and this is sleet, this is neither of those two. But at the time, there weren't good remote sensors for detecting exactly what was the precipitant. So my colleague Neil Cole and I designed an optical distrometer where we could tell the difference between, it was just a little laser thing that you could make for a few $100 to set it out in the Antarctic somewhere and take data on, like what's falling out of the sky, right? How fast and how big is it? From that you could infer, was it a snowflake or a water drop or whatever. And we proposed this to the NSF, and we got flatly rejected. That won't work, stupid idea, whatever. And these are supposed to be confidential, right? So just a few people saw that, okay? And I still, to this day, get emails from people who are, in Antarctica. They're like, how do I build that thing? And I heard all about it and everything like that. So these processes, they think, there's a grand total at maximum of like 2,000 people on Antarctica. And I'm not going to say I heard from most of them, but I've heard from a lot of them. And they want to know about how to build my optical distrometer. It was never published. It was put in one small NSF grant proposal that was shut down. And now for some reason, everybody thinks, that I've done this and published it and everything. So these processes of scientific review, merit review for funding are extremely leaky. And I only give that example because it's so extreme and so far away from medical stuff. I've probably had, I'm going to take a wild guess and say 6 or 8 really kind of important medically related things where that's happened, where I've either published something and it was rejected by the editors, and the next thing you know, they get a grant to do exactly what I had proposed. And in fact, in one case, they copied my paper so closely that it had the same spelling errors. Did I tell you that one?
No.
Oh, it was awesome. This was at Brown University. I'd love to name names, but I can't. So these guys actually this guy was an editor for a journal, took a paper that I had published, rejected it. A grant application I sent into DARPA was involved in that, rejected it, and then rejected other things I'd done, and then took my paper and submitted it to DARPA, and actually got funding for a paper of mine that he had rejected.
Yeah, but didn't he lose his funding? He got busted.
Well, he got busted because he was so lazy. He actually copied my typo, and then one of my grad students brought it to the federal government and said, "This is such an egregious case of scientific misconduct. You have to do something." So they actually did. They called up Brown University and said, "Send all of this stuff back." Put it all in crates. So, but the thing is, this happens all the time. I'm just like one little guy that nobody cares about, Why should that have happened to me 6 or 8 times? How many times has it happened to you, where somebody steals an idea from one of your papers and then takes credit for it, or refuses funding for you and then
I will say, we
give me a number.
I got so many rejections because everybody thought we were crazy 15 years ago, or on the brain gauge, and
it is
I just finally, but. Then, by the time people thought, that's a whole long story.
We're going to talk about this again.
About that again.
It's totally different because we're at the 1 beer limit here, but there are many cases in science, and this really matters to people, right, where you cannot publish a new finding until somebody else dies and leaves an opening on that editorial board or that review session.
Well, a lot of times it's sort of along the lines of, it's okay for one of the beautiful people to publish that,
not
but
me.
Not some grumpy old
guy. Well, if you're sort of, and so sometimes maybe legitimately, well, even that's not legitimate. I mean, if they have the same idea and they're working at it at the same time, they should not use their position of power to bias and hold back the publication or the funding of somebody who they're competing with. But they do all the time.
Yeah, just to give you another number to think about.
Hopefully, it's
how
not bigger than this.
how equally fair different study sections are. I actually have one grant that I submitted to one study section. This was the same study section that we rejected our stuff completely.
Mm-hmm. Wonder if I hear.
I got the worst score in the study section. It's very embarrassing to get the worst score.
Oh yeah, that's an accomplishment.
Without any changes. I submitted that grant application to another study section and made sure it went to the other study section and said, because, you know, I wrote a letter and said, well, I think these guys are sort of, you know, biased or whatever. And the people that were deciding which study section to go to kind of agreed with me. They said, well, okay, but, you know, they got the worst score. What's he going to do? He got the best score in the next study section.
So it tells you important things about NIH study sections, right? That their standards are so random that you can go from being the worst to the best in presumably the same level of scholarly, you know, vetting. It kind of tells you there's just a lot of noise in the system. So if you apply a little bit of bias in favor of you and your buddies and a little bit of bias against those people you're competing with, you know, it skews science. And there are whole fields of science that are held back for generations because of people that just won't let things through the funding and publication cycle. And that's a fact.
Well, I just got a text since that our guest victim.
We have a guest victim coming. So that's a completely different
stay tuned.
Video.
You're in for a treat.
You're in for a treat. We're going to increase the grumpiness in this room by 50% at least. All right. Talk to you later.